Hello everyone. I welcome you at Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, today I'm going to share my research and my knowledge under the title of Microbial Spoilage of Pharmaceutical Product. We manufacture different kind of the dosage form, tablet, syrup, injectables, and cream, ointment, suspension. These are susceptible for the microbial spoilage, and we are going to discuss such kind of the complication today. For this purpose, I collected the information and scientific research from different sources like PubMed, Medline, Scoopers, then Google Scholar, National Library of Medicine, MBAS, directly of open access journal, Duaj, ProQuest, CNKI, that is China-based database company, provides scientific research information all over the world. CNKI stands for China National Knowledge Infrastructure, some information collected from the cross-reference research field. Okay, I'm going to cover the content of under the three main headings: definition, susceptible pharmaceutical product, the dosage forms, and the preparation that are more prone to be contaminated and less prone to be contaminated by the microorganism. And last one, the microorganism involved in pharmaceutical trials. Okay, well, before moving forward, I would like to talk a little bit about the microbial spoilage. That is actually the damage to food or pharmaceutical product caused by the microorganism. That may be the bacteria, mold, and yeast, and they most of the time destroy and damage the pharmaceutical product. So deterioration of pharmaceutical product by the contaminated microbes is called as microbial spoilage. Microorganism can grow in almost all kind of the product, and microbe exist everywhere, all around the world. Therefore, a risk of microbial spoilage is always there. Pseudomonas species, yeast, and mold are more prone to contaminate the butter. Butter, high fat content, and low water activity make it more susceptible to the surface mold growth than to bacterial spoilage. Pharmaceutical products are prone to contamination. There are five different types of the pharmaceutical product or pharmaceutical dosage form that are more prone or microbial contamination. These products are the aqueous or the aqueous containing the product. Second one is the product with the neutral or the nearly neutral pH, product with the high nutrient availability, product with the low osmotic pressure, and last one the product with the high oxygen availability. These five kind of the pharmaceutical dosage form are more susceptible for the microbial spoilage. And six pharmaceutical dosage form or the product less prone to be microbial contamination. First one is the dry powder, then tablets, then hard gelatin capsule, product with the acidic or alkaline pH, product with the low or no nutrient availability or ability, product with the high osmotic pressure, for example, product having the high sucrose content. Okay, well, microorganism involved in the spoilage of pharmaceutical product. First one is the Pseudomonas aeruginosa. We know the Pseudomonas aeruginosa is gram-negative bacteria, and it is most of the time observed and noticed in the enteric, like in our intestine, in our GIT, gastrointestinal system. So it only stay outside the intestine. It most of the time don't produce infection in the mucosa, submucosa, or the membrane of the intestine. Now the next one is the Salmonella cavabon. It is a gram-negative rod that noticed and identified in the internal and external part of the intestinal system. It can produce infection in membrane inside and outside. The next one is the E. coli, Escherichia coli, that is gram-negative rod. It can produce infection inside and outside of the intestinal membrane. Okay, well, this is actually normal flora. It stay in the human body like commensal. Staphylococcus aureus, that is a cocci gram-positive bacteria, and it is aerobic. It most of the time produce infection externally on the skin and on external part of the body. So it may produce the microbial spoilage of pharmaceutical dosage forms or pharmaceutical product. Clostridium botulinum. This actually bacteria is gram positive. It is rod. It is anaerobic, and it produces infection in deep tissues because it may not need the oxygen for its metabolism. So it produces infection in deep tissues and produce the gases. The next microorganism that is able to produce the infection is the Clostridium perfringens, that is gram-positive rod and aerobic. You don't need oxygen, but they grow, reproduction and replication in body and produce the gases. The major complication of the disease produced by the Clostridium perfringens is gas gangrene. Gangrene or gas gangrene. The next microorganism that is able to produce the microbial spoilage is the Staphylococcus faecalis. This is gram-positive cocci and oxygenated bacteria, the aerobic bacteria that need oxygen for its multiplication, metabolism, and reproduction. It is also one of the commensal normal flora stay with the human body or human society or population. Next microorganism that may spoil the pharmaceutical product is the Proteus species. Proteus species belong to the Proteus providential Morganella, that is one of the group of the bacteria stay in human body like a commensal, like normal flora. It is able to produce inside and outside of the enteric system or the enteric layer. The infection, it means like it can produce infection in the mesenteric or the mucosal or submucosal membrane of the GIT. So this is also one of the challenging bacteria that produce complication in pharmaceutical product. Then the Pseudomonas species, that is gram-negative rod. It also stay inside and outside of the enteric membrane. Okay, well, the next group of organism or the bacteria that can spoil the pharmaceutical product are the gram-negative bacteria. In gram-negative bacteria, most of the bacteria belong to gram-negative rods. They may be the Klebsiella, maybe the Pseudomonas, maybe the Serratia, maybe the Proteus, then Providentia, Morganella. These are the potential challenge for the pharmacist working in pharmaceutical industry to save the pharmaceutical product from these organisms. So in conclusion, the microbial contamination of pharmaceutical product dispensed in hospitals or manufactured in industries pose potential threat. The Klebsiella, Bacillus, and the Candida species are predominant contaminants. The identified microorganisms like Bacillus species were resistant to augmentin and cloxacillin. Poor handling of pharmaceutical preparations or the product during the dispensing and or repacking contribute high rate of the microbial contamination. However, overemphasized and appropriate training on personal hygiene and the proper handling of medicine can reduce the spoilage. These are essential for the prevention and the control of the microbial contamination of pharmaceutical product. This is the reference from where I have collected the information today to present my information and the data. These are my colleagues and my pride, my team. They helped me a lot to make my video today. I would like to salute and greet to Thierry Bajkar, Wolfgang, Daniel, Dustin Hawker, then Marwa Al-Maghribi, Robert, Kunza, Noreen, Amin, Arslan, Ishtiaq, Faisal, Shahid, and Tarimil. Before closing the video, I also like to acknowledge and appreciate the kind help and support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development. Thank you very much for watching the video and staying with me for so long time. See you soon in my next video, and bye for now.